So Tom, thank you for that kind invitation. It was very difficult to turn down this meeting as I literally live about 3 miles from here. So there are not many chances you get to give presentations that don't involve airplanes, trains, or hotels. So it's a pleasure to be able to commute a very short distance to the meeting this morning. I'm going to speak very much with a bias of an oncology drug developer, and we'll follow up on some of the themes that Dr. Cleary mentioned earlier. And we are in a very, I think, dramatic time, both in oncology, in the changes in oncology, and in diagnostics. I'll talk a lot about the transition and the emergence of immune-based therapies as the backbone of oncology therapies, adding to the long history of targeted therapies, particularly tyrosine kinase inhibitors and signal transduction inhibitors, and the changes that that's going to require in diagnostic strategies to predict those patient response. But also, I think it's worth noting that over the last 3 weeks, I think the diagnostic field has really changed. Began with the approval of the Oncomine panel, and then more recently, as was mentioned earlier, the approval of the IMPACT panel, and then recently the Foundation Medicine panel. I don't fully understand all the implications of those approvals and why some, in some sense, some seem to be going down a PMA pathway, some seem to be 510Ks. I know there are many people here who understand the regulatory issues of that much better than I do, and I look forward to hearing about hearing the discussions about that. But I modified this slide, recently just because of this. But when we think about it, there are probably now almost 100,000 if you look at databases, they list about 100,000 biomarkers. But as of earlier this year, there were only 29 FDA-approved companion diagnostics, primarily in almost exclusively in oncology. And until 3 weeks ago, there were no multiplexed IVDs approved by the FDA. And so that number has gone from 0 to 3 very quickly recently. And if we think about the numbers of biomarkers, that's suggestive that there's roughly 4 biomarkers for every gene in the genome. So it's really become to the point now that the term biomarker has become too broad and too non-specific as to be really useful. Almost every gene, almost every analyte in some way is a biomarker. So why so few have developed, both the analytical validation for the test and the development of the clinical utility to deserve actually meeting the requirements for an in vitro diagnostic, and it is still a tiny proportion of the markers that we use. And then, why so few multiplex IVDs? And some of this comes down to this issue. Biobanking is a key part of it, the numbers of samples we collect in our clinical trials. In most cases, our clinical studies are simply not designed to develop and then statistically confirm complex molecular profiles. And the approved molecular profiles, like mammaprint, are all prognostic markers, and they were developed using biobank samples with 5 or 10-year known outcome, and large numbers of samples. And so if you think from a drug developer's perspective, we simply, one, you don't by definition don't have the long-term outcome data because it's a new drug. And secondly, you typically may only have sample numbers in the tens to hundreds, rarely the thousands. And so you simply do not have the statistical power to both develop and then independently test, complex panels. But I'll come back to that, in a minute. So so the question is, why would a drug developer want to use diagnostics? Well, the key issue at the end of the day is the most important thing around getting a drug approved is finding an indication, a predefined set of patients in whom there is a high response rate and the drug is highly effective. In oncology, we're able to build, we don't have it, basically get all of our approvals at once. We're able to build a drug indication by indication. And so the first approval really is often driven by finding a subset of a disease, an indication, where you have a high response rate and we can develop the drug successfully, and then we can then expand that, by subsequent indications. So in a recent survey that was published last year, it showed that, if you're looking, the probability of successful transition from out of phase 1, so at the beginning of your dose escalation studies, to a regulatory approval was about 1 in 4 if you have a predictive biomarker, and it was about 1/3 of that without a predictive biomarker. So that obviously, really speaks to the issue, to the point that if you have a good marker that identifies a subset of patients in whom the drug responds, your probability of success is enormously better. And this is even with all the work, the preclinical work, the work done on models, the genetically engineered models, the GEMs, the patient explants, and so on. We find that in oncology, only about 5% of compounds that enter phase 1 clinical testing actually emerge as regulated drugs. And somewhat disappointingly, that turns out to be about half the rate of the rest of the industry. So, if you look at all the other therapeutic areas, nobody else is down around 5%. They're typically around 10%. And this is sort of, I think, a little mystifying at first, because we have all these wonderful models in oncology. We can genetically engineer, we can knock down particular targets, we can overexpress targets, we can take patient explants. You think we understand the biology of some of these tumors quite well, yet we still only have a 1 in 20 chance of taking those targets forwards. And why is neuroscience, for example, better? I mean, a neuroscience model often consists of dropping a rat into a tank and seeing if it swims. And if it doesn't swim for very long, the rat is presumed to be depressed. Yet this model has twice the success rate of our sophisticated models in oncology. So it can't be the models, it has to be something else to it. And I think the issue is, and we'll see this going forwards, is there is a lower threshold for bringing compounds into development in oncology. We can show, because of the effect size of somatic mutations, we can show efficacy and safety, efficacy in particular, in a much smaller group of patients because of the effect size of these mutations. So if you think of a typical tyrosine kinase inhibitor, if you have an activating mutation, you typically will have a 30 to 40% chance of responding to an inhibitor of that activating mutation. If you don't have that activating mutation, you have no chance. So there is a big effect size. When you're looking at more complex germline-driven diseases, the diabetes of the world, the schizophrenias, the bipolars, you're dealing then with individual genetic variants that have very small individual effect sizes, and these become much harder to determine and require much larger samples. So I would argue that much of the work in oncology is driven by our ability to find these large effect size changes, driven by somatic changes, driven by changes in neoantigen recognition, as was mentioned earlier, that we have bigger effect sizes to work with so we can work effectively with much smaller patient populations. So when we begin to predict response to therapy, there are a number of things we have to have. And I think you'll see consistently one of the most important is a strong biomarker hypothesis. We have to have a very simple, typically up to now, these are typically measuring of the target status and emerging now more complex molecular profiles. But if you think of it, most of our successful diagnostics basically measure the mutational status of a target in signal transduction, or the presence or absence, by the presence or absence of particular mutations or translocations or copy number changes. We have to have a large number of correlative endpoints that we can then correlate to those, to those endpoints. These can be SNP panels, it can be gene expression profiles, it can be mutational, whole exome, whole genome screens. One of the difficulties we face in oncology is our endpoints are not consistent. When we begin the process, we're looking for compounds that kill cells or shrink tumors in models. When we move into regulatory testing, we're now basically measuring survival. So, and we do know now that predicting response is not necessarily the same thing as predicting survival. Typically, something that has very high response rates has a much higher probability of showing a survival benefit, but it is not a clear correlation. And then also, there's not a clear correlation between the different survival measures, or whether you're using progression-free survival or overall survival. And lastly, as I mentioned earlier, you had to have large numbers of samples in order to be able to have training sets to define your models, and then fully independent test sets to independently test those. And we know from experience that when you don't properly separate your training and your test sets, you can lead to overfitting of data and self-fulfilling prophecies. So if you look at the history of oncology drug approval since 1998, that was the year that Herceptin, the first oncology drug with a companion diagnostic, was approved, now almost 2 decades ago. And you look at the numbers of drugs that are approved with and without tests. This was a publication for earlier this year. The light gray is drugs approved with a test. So in the 1998 column, you can see that light gray bar is Herceptin. The dark gray represents drugs that were approved by the FDA without a diagnostic test. So in essence, the developer was able to show sufficient activity in an indication in order to justify approval. And if you could look, even up through 2016, you can see that the majority of oncology drugs are still being brought to market today without a companion diagnostic test. And in 2017, that trend continues. Obviously, there's some more markers. Perhaps the most important change is what was mentioned already, is the approval now of these multiplex tests for the first time. The Oncomine test is a 23 gene test, and then 2 tests an order of magnitude bigger, the Foundation 1 test, and then the MSK Impact test, which are 300 and 400 genes respectively. I think it's worth pointing out that these tests are very different to a test like a mammoprint. Because in essence, what these tests do is they take single analyte results and single analyte tests and combine them into a single sequence analysis. So you're testing a BRAF status, and you're interpreting the decision to give a BRAF inhibitor independently of the other data in that panel. You're looking at EGFR, you're looking at KRAS, you're looking at ROS1. Whereas if you look at the tests like mammoprint and some of the other more complex tests, they are really taking all of the analytes, interpreting those with a black box algorithm, and then giving a unitary result. So I think we have to think about these tests in very different ways. These tests all combine things that could be done by individual single gene IVDs, but for convenience, include them into a single test. The other tests basically take multiple data points and use a complex algorithm to infer a patient's status from them. And I think we have to view these tests quite differently. So some of the common features, I mentioned some of these: strong hypothesis. The predictive marker test has to be available very early in the development. Typically now, we're seeing with oncology that the old model of a phase 1 dose escalation to early signal activity testing in phase 2 and a registrational phase 3 trial is really gone away. We're now seeing typically now drug approvals in less than 3 years that basically involve a phase 1 dose escalation, a phase 1 in an unsegmented population, so essentially all comers, upon the definition of a safe and effective dose, a phase 1 extension only in the particular mutant carriers, and then a registrational randomized trial immediately after that. And we now have numerous examples of drugs with diagnostics approved in less than 3 years. And so clearly, the argument diagnostics accelerates drug development. It does not slow it down. The issues of biobanking and other things do add some cost, but I would argue the balance is so much in favor of having the high activity that this is clearly a universal approach to way drugs will be developed in the future. So the other big change that those of us in oncology are facing is the emergence of immune-based therapies. And this is something that has happened over the last few years, last really 5 years or so clinically. And it has changed the way we think about treating cancer. For most of my career, I've worked on drugs like tyrosine kinase inhibitors, and I remember how difficult it was to develop them early on. and now we can do them very routinely and quickly. I think immunotherapies are very much in the early stage, and we are, in some sense, running a little bit before we can walk, because we've seen some extraordinary results with things like the CAR-Ts and with the checkpoint inhibitors, but these have been hard to broaden and hard to replicate in all cases. But we do have now the premise of extraordinary long-term survival, essentially cures of subsets of patients using immune-based therapies, and combinations of treatment with immune-based therapies, and cures with longitudinal, robust survivals that we've never been able to achieve therapeutically with targeted signal transduction. But this is going to require a very different approach to the development of diagnostics. So if we look at the approved list, and I apologize, this list may be 1 or 2 drugs out of date. The vast majority of the approved companion diagnostics are for targeted therapies, the TKIs, essentially measuring signal transduction, DNA repair deficiency, functional things like that. Other than, we are still in relatively early days looking at predicting response to immunotherapies and looking at things like the expression of the PD-1 ligand, T-cell infiltration, T-cell clonality, and various things, and these are still beginning to emerge. And again, looking back at chemotherapy, we've never been good at predicting, response to chemotherapy and understanding why some people have more prolonged response than others, and it's clearly a multiplex, complex situation that we just simply can't predict accurately at the moment. So if you think about the decisions we now have to make, traditionally, if you look at the left in blue, the key question we were making from a diagnostic perspective around targeted driver mutations was, is that activating mutation present in a patient? If so, you have a clear clinical hypothesis that you inhibit that pathway, the patient will then clearly benefit in terms of survival, disease delay and disease progression and survival. But if that mutation is, so for example, if you have BRAF wild type, it is presumably futile to treat with a BRAF inhibitor. The key question we have to ask now in immune-based therapies is there a prior immune response? Because in the same logic, you would not want to give a checkpoint inhibitor to a patient who has not had a prior immune response, because there's no immune response to release. Those patients now need to go into an alternative therapeutic strategies of trying to boost and drive new immune responses. But to give a checkpoint inhibitor to a patient who does not have a prior immune response is a waste of everybody's time, because there's no, that cannot work, because there is no immune response. There's no checkpoint to release. So how do we do that? So I think in that sense, we really have to basically look, the 2 basic ideas is if there is evidence of a prior immune response, then the therapeutic goal is to release that immune response that has been inhibited by the tumor and allow the immune system to try, to impact on the tumor. If there is no immune response, then we have to use new approaches, whether it's vaccines, T-cell modulation, T-cell redirection, ex vivo modification, CAR-Ts, so on, to induce a new immune response. But these things are likely to be quite separate. So how are we going to do this? I think, so when we think about the data that is emerging now around the checkpoint inhibitors, is the patients who have high infiltration. So if you look at the tumor and you see large numbers of T cells having infiltrated their tumor, if you then sequence their VDJ genes and you look for clonality and you find a large number of clonal T cells in the tumor, that is by definition, that is almost the definition of an immune response. You've had a clear clonal T cell expansion as a response to a particular antigen. So here, this is almost a textbook example of how a T cell will respond to a tumor. We find that those tumors that have high expression of the checkpoint, the inhibitor of that immune response, also are likely to respond. So you have a combination. Those patients typically have a higher mutation burden. They carry more mutations. And as we begin to understand what was mentioned earlier, this ability to predict which mutations are going to be presented by the MHC and become detected by the immune system is starting now to be revealed. But what we see from some recent publications that came out over the summer, it seems like a lot of our driver mutations are located in peptides that are invisible to the immune system. They are not presented by the MHC. So they will never, so you could argue that the driving mutations perhaps are not driving mutations. They are just symbols of survival of the immune system. Which is a completely heretical idea, but it's one I think we need to explore in more detail, because it seems very clear that again, using a BRAF or an EGFR, these mutations do not occur in peptides that are presented by the MHC to the immune system, and it seems like almost all of our canonical driver mutations occur in peptides that are not presented by the MHC. So here with checkpoint inhibitors, the classic deactivate, the PD-1, the ligand on the tumor cell binds to the PD-1 receptor on the T cell and inhibits the T cell activation. An activated T cell, and so essentially suppresses, so you then, by using a checkpoint inhibitor, you essentially block that suppression. So when we're predicting response, we can see high expression of the ligand. On the top right, as you're seeing, is the evidence of T cell clonality. This is data from published in Nature, again, which is showing a preponderance of clonal T cells in the tumors that are highly immunogenic. And then also, the nature, the frequency of the underlying mutation burden. So typically what we see is, we've known I first started my career in oncology working on melanoma in an immunology lab. We had no idea why it was the most, immunogenic of tumors. We now know it's because of the numbers of mutations. Mutations in melanoma are created induced by ultraviolet irradiation. Ultraviolet irradiation of DNA leads to the formation of CpG dimers. And then and if you have an intact DNA repair mechanism, those CpG dimers are cleaned up by essentially 2 base excisions. So melanoma sequences are full of 2 base pair deletions, or essentially frame shift mutations. When we look at microsatellite instability, Luis Diaz's and others' work, basically, again, you see huge numbers of mutations if you have deficient DNA repair compared to very much smaller numbers of mutations. So this is a sort of stochastic model. The more mutations you have, the greater likelihood of one of them being picked up and seeded by the immune system. Again, so this is the data, well-known data on Pembro, but again, with the mismatch repairs. So here in the Diaz paper, a typical mismatch repair patient has 1,700 mutations compared to the normal. If you have normal mismatch repair, it's less than 100. So huge increases in the number of mutations. So again, this sort of speaks to this stochastic chance that if you have mismatch repair, that you're going to have a much higher tumor mutation burden. If you have a higher tumor mutation burden, you have a higher chance of producing an epitope that will be recognized by the immune system. And then this is just some of the survival, just to save time survival data. So now what do we do with patients if you don't have this prior immune response? Giving a checkpoint inhibitor is not going to do any good because there's no immune recognition to release. I saw a presentation recently where somebody compared this to the problem a zookeeper has in the zoo. So if you could leave the tiger cage open if there's no tiger in the cage, but if there is a tiger in there, there's a serious consequences to leaving the door open. You think of the checkpoint inhibitors much the same way. There's if there's no tiger there to come out, if you inhibit the checkpoint, but if there is, there's a response. And I thought that was really quite a good analogy. So what do we do now? So what are the approaches now? And I think probably, and then just some of the examples from our portfolio of approaches we're taking to try to induce immune response in these otherwise immunologically cold tumors is the development of antibody-mediated cellular cytotoxicity. So here essentially, you're very much dependent on a protein-based test, an immunochemistry test, to looking at the target cancer antigen. So this is not going to be driven by a DNA test. It will not come from a DNA sequencing panel. in the antigen you're picking to drive that ADCC response. It needs to be highly expressed on the tumor, minimally expressed on critical normal cells and tissues. And this is essentially an immunohistochemistry test to drive the selection of proteins you may use for ADCC responses. T-cell redirection is this idea where you take a modified antibody and you make essentially a bispecific molecule. So imagine, you think of it of an approach where you're essentially gluing two antibodies together and cutting out some of the middle chunks. So you have a protein molecule that will recognize and bind to two different epitopes. So in the case of T-cell redirection, and there are a number, there are a whole lot of different approaches. This example is a BiTE, but there are dots, there are duobodies, there are probably a dozen of different molecular constructs of these bispecific markers. But the general idea is you have one end of it binds to a ubiquitously expressed marker on the T cell, a CD3, for example, and then the other binds to a protein expressed on the surface of the tumor. And here in some in vivo models using a duobody in this case, so again, it's still a bispecific, and then with active at both ends, and then in the null control, sorry, let me go back. In the null control, you only have one active end. So the duobody has, I forget if it's active to the CD3 or active to the tumor end, but only one of the ends is active in the controls here. And you can see over time that with the null control, you do not increase the numbers of T cells present in the tumor. But here, staining for CD8 positive T cells, you can see progressively over time a significant increase in retention in the number of T cells in the tumor, driven by the presence of this active duobody. But again, we need here, this is now a protein-dependent assay. You're not going to be able to identify which are your optimal patients for particular targeting from DNA sequence. Vaccines, another approach. So if you're trying to induce an immune response with a cancer vaccine, then the issue is going to be, so this is an example of HER2 expression in a breast cancer, but we have to be able to again, look at the protein to understand which particular protein is being highly expressed on the surface of the tumor, because there's no point in vaccinating a patient with an epitope that is now not present on the tumor. So again, just to make the point, all of these non-checkpoint approaches required to develop new immune systems are not DNA dependent other than the crude tumor mutation burden test, but are going to be dependent on specific protein tests. So just to wrap up here, I think what we're seeing with the emergence now, with the change in the FDA strategy around approval of complex diagnostic tests, this is the beginning, I think, of a very exciting era for diagnostics. I think we do need to remember these tests are just an accumulation, essentially, of individual IVDs. They are very different to the tests that require sort of complex black box algorithms to interpret multiple analytes and draw a single combined conclusion. But it's clearly a step in the right direction. Immuno-oncology is here to stay. These long-term survival data, the anecdotes we hear are all true, and they're becoming increasingly common. You have patients like former President Carter diagnosed with metastatic melanoma with metastasis to his brain and his liver. This is usually a diagnosis that is not compatible with long-term survival, and I think he's now more than 5 years out post-diagnosis, still disease-free. This is a miraculous response, and we need to learn to make that ubiquitous, not rare. And this will drive that. But we're going to need to think about, so those of us in the business of developing drugs with diagnostic tests, we know we can't rely on DNA-based testing alone. We're going to have to look at how we measure the interaction of the tumor with the immune system in real time. It will not replace DNA-based testing, but it will be supplementary. But the biomarker assay development is only going to be more complicated going forwards than it was for the traditional tyrosine kinase inhibitors. So I'll stop there. Thank you for your attention. I'd be happy to take questions if there are any time or at the break.